patient 3, please come in.
Okay. Okay, I'm here.
Okay. Sit down. 1 second, let me grab a mask. Hello, how are you?
Not good. I've been having a terrible cough that seizes my lungs. I feel like there's a brick in my lungs. I've got a fever and I'm so tired. I've had the flu and COVID test, and I have neither.
Oh, okay. Oh no. Have you been in contact with any mice recently?
Well, yes. I have a house back in the forest, so I see a lot of deer mice.
Deer mice? You could have the hantavirus. You could die.
What's that?
It's an envelope glycoprotein spherical virus found in certain mice and bats. While it's rare, it still occurs. You have lots of deer mice by you. You need to get to the hospital. Its genetic code has already been injected into your cells, especially your lung cells. You could get really sick really soon.
Wait, I have cells? What the heck are those?
Here, take this medicine and sit while I explain.
Okay.
Okay, so let's talk about cells. Ha! You thought I was going to actually make a successful whiteboard video? Well, you thought wrong. Or at least my phone did. Well, you see, my phone decided that at 12 o'clock at night, it was going to stop recording, and it refused to record. So, I don't have much to work with other than a bunch of slow motion videos that I don't want. So, I'm gonna take a different turn on this and make it funner. Funner for you to grade, funner for you to make, and it'll still have all the information in it. It'll be a little bit more chaotic. So let's try this. Okay, back to what I was saying. Cells. The cell theory is that all living things are made up of one or more cells. Yup, you. And that they're the basic unit of structure and function of all organisms. All cells come from a pre-existing cell. Yeah, that's why you're not a, clump of dead cells. But we'll explain that later. The two different types of cells are prokaryotic and eukaryotic.
How can you tell the difference between the two basic types of cells?
Well, this dying human is pretty smart. Prokaryotic cells, which are the ones that are shown here, they lack a nucleus and membrane-bound organelles. Prokaryotic cells are overall smaller, have single chromosomes, the DNA is circular and unbound to the protein. There's no membrane and about 70 ribosomes. Eukaryotic cells, which are the human cells, have a nucleus, go through mitosis and meiosis, are larger, contain a nucleus, are membrane-bound, have linear DNA, and have 80 ribosomes. Let's just say you want to be a eukaryotic cell, so you're not a parasite or a bacteria, which is what they commonly are, walking around basically with no thoughts. Actually, let's not give these cells any ideas because I don't feel like being a zombie today. Anyways, eukaryotic cells are in things like animals and people.
What is a cell membrane?
Okay, a cell membrane is often made of a phospholipid bilayer. Wait, not often, always. Why did I write often? Anyways, it is always made of a phospholipid bilayer. It is made of a phospholipid. On the outer layers, there is a hydrophilic head that likes water and a hydrophobic inside layer that dislikes water and repels it. Throughout it, there are channel proteins, carbohydrates, and cholesterol throughout the phospholipid bilayer to assist the semi-permeable membrane to pass substances needed and not needed in the cell. I wish I could do that with healthy and not healthy foods, but I like food.
How?
So, how is very misleading and pretty general, but I'm guessing you're asking, what ways the cells move materials across the cell membrane, or like how. So I'm gonna say cells do this by channeling proteins which distribute ions and molecules across the cell membrane based on the concentrating gradient on their side. So like, for example, if you have a farm and you need sheep, you have lots of sheep in your pen, okay? But there's only some pigs. So you need more pigs in your pen, okay? So you're obviously gonna take the gradient. So if you have more, you need more of what you have less of. So that's how I'm gonna explain the concentrated gradient. But if you have an unequal one, so let's say you for some reason need more sheep on your farm, you can, oh, this is very bad. Okay. Charged channel proteins using ATP are used when molecules or ions are nonpolar and large or have a low to high concentration. So if you need more sheep for some reason, but you already have pigs and you can't physically fit any more sheeps in there, you can like use some more energy that you don't usually use and shove all the sheep in there. If that's a good yeah, let's continue.
What is a cell cycle?
This is a long one. The cell cycle is how your cells come from pre-existing cells. So the cell cycle is a series of events that take place in a cell for it to divide and reproduce to two daughter cells. And before your coughing sick self asks what those processes are, I'll just explain. So the cell begins an interphase where it spends most of its time. There are G1, S1, and G2 interphase stages. In G1, the cell begins DNA replication, increases in size, and stores nutrients. In S1, each of the 46 chromosomes is duplicated by the cell. DNA replication finishes. During G2, DNA synthesis occurs, and the cell double checks for any errors or repairs that may need to happen. So like if you're building a car, and like you really need it to run correctly so you don't kill someone, you want to make sure everything's good. So that's what they're doing in interphase. So now it moves on to prophase, where the nucleus disappears and the mitotic spindles in the front of the centrioles begin to reach out to the chromosomes. After prophase, there is metaphase, where the spindle fibers attach to the midpoint of the chromosomes. So next anaphase occurs, where the fibers pull apart the chromosomes and bring one half to each separate side. So it kind of makes sense. You have these things in the middle that are used in DNA replication, and they're very important to the cell, and now they're being equally pulled apart to each side. So now telophase and cytokinesis follow. They're generally similar, but there are a few differences. In telophase, the nuclear envelope forms around both sides of the chromosomes at the midpoint, so it like starts folding. And cytokinesis is where the envelope fully snaps and pinches and separates into two daughter cells. So do not confuse them.
How did I get sick? How is it spreading?
How did you get sick and how is it spreading? Well, other than the obvious fact that you are living with deer mice, which is extremely risky, I'll go the scientific route, just to confuse your sick state more. Where human cells are eukaryotic, there's orthohantavirus, which is a prokaryotic cell. You remember our virus that we talked about before? Yeah, scary. Hantavirus is a viral lung disease, which is a single-stranded envelope virus that is found in bats, rats, and rodents. Has a spherical shape. Human cells start with a stem and go in different shapes and sizes based on the type of cell they are. Hantavirus infects the human lung cells by their outer glycoproteins that stab into the cells and inject their DNA, forcing the cell to produce needed materials for the virus and taking control of the cell, which reproduces more viral cells. By the way, I cannot read, and it is currently 12 o'clock at night. Just appreciate the fact that I'm still awake. Hantavirus early symptoms are general and include fever, fatigue, muscle pain. More severe cases can cause shortness of breath, fluid in the lungs, and death. The coughing is concerning me. Inside hantavirus cell, there are 3 helical nucleocapsid strains. Whoa, some big words. They are small, medium, and large. On the outside, the spike proteins are glycoproteins that have 2 sizes, G1 and G2. I'm pretty sure I already said this, but G2 is smaller. So yeah, cells are unique to the type and replicate quickly for good and bad. What are you doing?
I said walk over.
No, I said.
Oh, can I breathe?
No.